Hello, everyone. Today's topic is viruses. This is a short chapter from your textbook, OpenStax Biology 2nd edition, and my lecture is going to cover most of the chapter. There's 4 sections, chapter 21, sections 1, 2, 3, and 4, and this lecture is going to cover sections 1, 2, and 3. Section 4 is about prions and virioids. If you're interested in reading about those, then you can go ahead and read that section, but it won't be covered and you won't be tested on it. So this picture is showing comparison between artist's rendering of the virus that causes COVID-19 and the virus that causes influenza. So they're represented as being these kind of circular entities with these little colored things sticking up from them. So by the end of the lecture today, you should understand what these images are showing and some of the characteristics of these little things that are sticking up, the wide variety of different types of viruses, and some more about how they infect us and how we can prevent ourselves from getting sick. So the formal outline for our lecture today is we're going to start by defining viruses. What are they? Talk about how we categorize viruses based on their structures, and then also based on a formal system that looks at what their genome is made of and how they copy it in the cell. We're going to look at how they replicate, which basically is part of how they infect the cell, how they multiply the structures that they need in order to make new viruses, how they get out of the cell again. And it turns out there's an important difference between what happens in animal viruses. So we'll use an example of the influenza virus and what happens in bacteria that are infected by a type of virus called a phage. Right, so we'll separate what happens in animals versus what happens in bacteria. We'll look at what viral infections can do to the body and why, and look at the variety of different kinds of hosts, how a virus might spread from one host to a different host, what types of transmission are possible, and end with a really quick discussion of prevention and treatment, although we will come back to some of these topics when we cover the immune system later in the semester. So hopefully you'll have lots more questions than answers based on what we cover today. Now the pictures that are on this slide are electron micrographs. Virus particles are very small. Prokaryotic cells, bacteria and archaea are on the order of 1 to 10 microns, right? Viral particles are on the order of 50 nanometers or so. So this little guy here, this phage on top of this bacteria is really teeny. So we didn't discover viruses until scientific methods had developed well enough that we could have the technology to detect such small particles or the evidence of small particles, and we couldn't image them until electron microscopy was invented. So these are very small, not organisms, things, right? And that distinction is important because viruses are non-living entities, right? We can't say they're a cell, we can't say they're an organism. If we go back to our definition of what things are living things, I gave you at the beginning of the semester the CHARM characteristics, which are complexity, homeostasis, adaptation, reproduction, and metabolism. Something has to be able to do all 5 of those things to be considered a living thing. It also needs to be made up of at least one cell. All right, so viruses are not cells. The basic concept of what a virus is it's a protein capsid or capsule that encloses some genetic material. Right, the genetic material is made of DNA or RNA, so nucleic acids, just like full-blown organisms have. And viruses also have complexity. They have a specific structure. They are built, each type of virus, based on the type of genetic information, the genes that are coded for in its genome, will build a certain structure that is complex in that it has an organization and a pattern. And so they do have complexity. They also have the ability to adapt. So even though they're non-living, because their instructions come from genetic material, just like living things, there can be mistakes in copying the genetic material that causes changes to the characteristics and how they are expressed. And natural selection acts on viruses just like it acts on living things, that those individuals that have a mutation that makes them better able to survive and reproduce are going to increase in the population, right? So we do see adaptation among viruses, but they cannot reproduce, carry out metabolism or homeostasis on their own. Right? Basically, a virus on its own is an inert particle. It's doing nothing. It is just existing. When it bumps into the appropriate type of host cell, it's going to bind to that cell. It's going to insert its genetic information into that cell. It's going to take over the cellular machinery. It's going to use the energy, the metabolic resources of the cell. It's going to use the ribosomes of the cell, and it's going to replicate its own genome. It's going to build more viral protein capsules in order to reproduce itself. Right, so we say it's an obligate. That means it has to depend on living organisms to reproduce, and that it's an intracellular parasite. Right, so it goes inside cells. It doesn't live on the outside of cells. It has to go inside. Parasite meaning that it is closely associated with the cell that it's attacking. And we talked about in our ecology unit, symbioses, types of interspecific interactions. We're going to extend that to these things that really aren't even species, but they're going to have a parasite-host relationship, just like we think about a flea being a parasite on your dog, the host. Because they're non-living entities, they do not have a scientific name. So we think about those Latin binomials. Bacterial diseases, on the other hand, do have a scientific name. Yersinia pestis is the plague causing bacteria. Yersinia is its genus. We write it with a capital letter, pestis with a small p. All right, but we don't name viruses that way. So this slide is showing an example of how we name different viruses. So you might have heard of the H1N1 flu. It's an influenza virus like this one. It is officially designated with the virus type, which is classified based on the type of nuclear material, geographic origin, strain number. If you've been hearing about COVID strains, we had alpha, beta, et cetera, Omicron. Sometimes they're lettered instead of numbered, the year it was isolated or discovered. And then the specific version of the virus subtypes. So influenza makes a couple different surface proteins that are sticking out from it. So those red sticks with the balls on the tip, those are representing a molecule called neuraminidase, and the blue sticks are representing a molecule called hemagglutinin. And these are important in how the virus attaches to and infects the host. So these influenza viruses always have these two types of molecules sticking up from them, different versions of these molecules with different mutations in them. So they have slightly different structures, have been numbered. So we know which strain of the flu virus this is. All right. When we look at all the different kinds of viruses, we find they come in 4 general structures. They can have a helical structure, sort of like a long thin rod with a little spiral of these proteins on the outside. So these protein coatings on the outside are built of little subunits. So the overall virus is called a virion, or a particle. Its external coating is called a capsid. And then the capsid is made of capsomeres. All right, meres are these little repeating units that are assembled together in order to build that capsid structure. So inside the capsid is the genetic material, whether it's RNA or DNA. It may have these glycoproteins like hemagglutinin sticking up from the surface. And so polyhedral or icosahedral is another shape. They look sort of geometric from the outside. There's another type that is going to be encapsulated or enveloped. Think about being inside an envelope. And that envelope is actually made of cell membranes stolen from the host cell. So it's going to be a phospholipid bilayer, and these are going to look spherical. So COVID and COVID-19 and the influenza virus are examples of encapsulated or enveloped spherical viruses. That is a type of structure that they can have. Anything that doesn't fit into these categories is called complex. Bacteriophage is an example, although there are other types of complex viral structures. I love bacteriophage. They look like little spaceships. They've got the head part, they've got a tail, they've got these tail fibers that look like the legs of some kind of landing craft. It's going to sit down onto the surface of a bacteria, and it's going to inject its genetic material inside that bacterial cell. So when we can look at the outside structure of these different types of virus, but that doesn't necessarily tell us what kind of organism it's going to infect or how it's going to go about doing its replication. So different strategies in naming and describing different types of viruses have been sort of used at different times in their history. And the one that we use today is based on the type of viral genome. All right, so when we look at the viral genome, this is an example again of influenza. We've got what genes it has over here on the left-hand side, and what they code for, what they make in this cartoon on the right-hand side. And viral genomes can be made of DNA, or they can be made of RNA. They can, either one of those can be single-stranded or double-stranded. They can be linear, which means that they stay in a line, or when they go inside the cell, they can form a circle like bacterial genomes do. And they can be in just one continuous piece, or they can be in little sections or segments. All right, so we have a segmented genome that's shown in this picture. And it's kind of like how our DNA is in multiple chromosomes, right? So they're separate molecules from one another. So it doesn't have very many genes. There's no shared genetic information between all viruses, so we can't necessarily do a nice taxonomy of them the same way we do for real organisms. And the only genes they have are the ones that they need in order to replicate their own genetic material. They'll use host enzymes if they have them, but some viruses do things that our host cells can't do. So the viruses bring their own. So they'll make enzymes in order to do things they need to do, including the viral polymerase. So this viral polymerase, all the stuff that's inside this cell, these are enzymes, and they assemble together the capsid for the virus to be able to reproduce itself once it gets inside that host cell. So it's going to make the proteins that it has on its surface, the hemagglutinin and neuraminidase, and any other proteins that it needs that might make up the envelope in this case, or the capsid. And so they have a small genome, and it only makes the proteins and enzymes and other structures that the virus needs to build itself and to replicate its genome. So the classification system that includes all different virus types, that's sort of the most modern and thorough, is called the Baltimore classification system. And it has 7 groups. And the groups are defined by looking at the very top of this figure. There's a lot in here. We'll spend a couple minutes on it. DNA, the purple on the far left, the RNA, and RNA that uses, ones that use reverse transcriptase. So reverse transcriptase is an enzyme that's only found in viruses. And when we think about the central dogma of how DNA is used in the cell generally, we have DNA that's stored in the nucleus, it's double-stranded. When a gene needs to be used, the DNA splits apart. The message side of that DNA molecule is copied into a messenger RNA using an RNA polymerase enzyme. That messenger RNA leaves the nucleus of the cell and goes out into the endoplasmic reticulum, where it's going to find a ribosome that's going to make a protein from that messenger RNA. So where we have viruses that use those same mechanisms, they don't need to bring any extra enzymes with them. But we have these viruses that use reverse transcription. Reverse transcription is something that doesn't happen in real living things, cellular organisms. It's making an RNA from a DNA. So it's an enzyme that's going to be able to make a DNA molecule from an RNA molecule. All right, so they have to have this reverse transcription enzyme. All right, so DNA viruses can have their genome that is double-stranded or single-stranded. So little d, little s means double-stranded. You can see it in the cartoon there with the double helix, both sides. Single-stranded DNA. RNA viruses can have double-stranded RNA, right? That doesn't happen normally in living organisms. They can have single-stranded RNA that's plus. That means it's the message that the messenger RNA would normally be made, how it would normally be made. So it's essentially the same thing as a messenger RNA. And then there's the single-stranded RNA minus. All right, so then we can also have these reverse transcriptase enzyme using ones that have a single-stranded RNA or that have a double-stranded RNA. All right, so each of these different types is going to have something it needs to do in order to make that messenger RNA. Double-stranded DNA is going to be used just the same as regular double-stranded DNA is to make a messenger RNA. Single-stranded DNA is going to use the DNA copying mechanism that's inside the cell already to make a double-stranded DNA. So it's just going to do like DNA polymerase, use DNA polymerase to do that, and then it can make a messenger RNA. Double-stranded RNAs are going to basically do the same thing. They're going to use RNA polymerase to make a messenger RNA. These RNA plus viruses may need to copy themselves into an RNA minus in order to make an RNA plus. They can also to copy the viral genome. They can also be used as a messenger RNA. The RNA minuses are going to be able to be used as a messenger RNA. The reverse transcriptase using viruses are going to make double-stranded DNAs, and they're going to use the normal cell processes once that happens. All right. So if you have already taken your cell and molecular biology, hopefully this made a lot of sense to you. If you haven't already taken your cell and molecular biology, I've got one of these examples zoomed in a little more closely. And you might want to go back in your textbook and look through the steps of DNA copying or DNA replication, translation, and transcription, if you haven't studied it before, in order to make sure you understand what's happening in all these steps. Right, so once a virus attaches to a host cell, it starts an infection. What it has to do is two things. Copy, just make bunches of copies of its viral genome to go into the new virions that it's going to make, its offspring. And it also needs to make all the different, needs to implement its genes. It needs to make the proteins that are going to make up the capsid, that are going to be in the enzymes that it needs. And so it has two jobs to do. It has to use the molecules of the cell itself and any proteins that it brings with it. So sometimes they're included in the capsid. It's going to make proteins, right? And it's also going to replicate its genome. Okay, so here we have an example of a single-stranded negative RNA. It's going to be able to transcribe that into the viral messenger RNA, and then that gets translated into the proteins. It's also going to use cells copying mechanism, RNA polymerase, to make single-stranded positive RNAs that then is going to use the same copying mechanism to create copies of the viral genome, right? So the goal, the purpose of all this copying is to produce all the structures that the virus needs, because what it's trying to do inside the host cell is make a bunch more copies of itself, right? It's trying to reproduce. All right, so let's talk a little bit more about the ins and outs of how the virus does replicate or reproduce. Right, there's 4 steps: attachment, entry, replication and assembly, and egress. Attachment is sticking onto the outside of the cell. It has to recognize and bind to the host cell. So each virus has specificity, or the ability to attach to certain types of cells in certain hosts, right? So every virus has to bump into its particular species of host and the right kind of cell inside that host for it to be able to bind. It's going to have either glycoproteins like hemagglutinin sticking up on the surface on this HIV virus. It's got this GP120, some kind of glycoprotein molecule, and that's going to bind to these CD4 proteins that are on the surface of some types of our immune system cells, right? So the CD4 is considered a receptor on the surface of this cell. It has a different purpose. Its life goal is not to stick to viruses, but the virus has evolved to have a protein on its surface that binds to this particular molecule, right? If it's not an enveloped virus, then other types of proteins are sticking up from the capsid, or other glycoproteins sticking up from the capsid. So these receptors and the glycoproteins, the attachment process works like an enzyme and a substrate. It's a lock and key. They have to fit together. And so we tend to have this very close specificity between a certain virus and the species that can be its host. All right, once it's bound, it's going to have some way to get inside the cell. Viruses with an envelope tend to trick the cell membrane into doing endocytosis to bring them inside the cell. They may, like bacteriophage, they may simply inject their genetic material inside the cell, but a lot of them are going to have the whole capsid get moved inside the cell. So they either leave the capsid on the outside or they bring the whole capsid inside. Then the capsid disintegrates and the viral genome and whatever proteins might be inside of it get released into the contents of the cell. Then it's going to use the energy of the cell. It's going to use proteins that are in the cell. It's going to use enzymes that are in the cell in order to replicate and the proteins that it makes to assemble new capsids with the genetic material. So once it's made a whole bunch of offspring, then they need to get out of the cell in order to infect new cells, right? So the way that the virus population is going to keep growing is there's only so many virus particles that can be made in any one cell, so it wants to spread to new cells inside the same host. It also wants to spread to new hosts, or new individuals. So this egress is the exiting, right? It's got to get out of the cell. Different types of viruses are going to do this. Sometimes they'll do it one way, sometimes they'll do it another. They have kind of a life history strategy where they may switch strategies depending on the condition the host is in. But generally, there are some types that typically are going to exit the host cell without killing the cell immediately. They may kill it slowly, eventually. Typically, most viral infections are going to kill the cell, because they're going to use up all the metabolic energy that it has. They're going to exit the cell. They're going to take a little bit of its membrane with it. So eventually it'll, its health will be compromised no matter what. Some viruses are going to replicate a whole bunch, and then sort of explode out of the cell, disintegrate again in the process. Others are going to leave sort of one at a time, a little more gently. We'll see some examples in the next couple slides. All right, so starting with animal cells and influenza virus, the way this looks step by step is that we're going to have attachment. We have the receptors on the surface of the envelope are going to bind to the surface of the host cell. The cell is going to engulf the virus by endocytosis, so it's going to come inside. It's going to be surrounded at first by host cell membrane. That's going to disintegrate. The capsid is going to disintegrate. It's going to release all these proteins, all these little bits of RNA into the nucleus. It gets copied by the mechanism that already exists in the nucleus. The viral messenger RNA is used to make viral proteins. And the viral particles, or the virions, are going to be released, assembled into new virions. And then the process that this influenza virus leaves the cell is called budding, and it uses the cell's own sort of exocytosis process in order to leave the cell. So the cell isn't killed by this and can continue to make new virus, right? So this is one strategy, and different viruses have these different strategies. If you want to think about the evolution of viruses, if you explode the host cell that you're in, it can't keep making new viruses. So there's a trade-off, right, between these different, sort of how long the host cell lives, how severe the infection is within a single host, right? How sick the virus makes the host. If the virus killed the host right away, that wouldn't be great for the virus because it wouldn't have a chance to get to a new host, right? So viruses are like weeds. They're going to try and grow really big population rapidly. They're going to be our selected life history strategy, trying to increase really rapidly, and then go to a new location, right? So they're going to want to disperse, and that means being transmitted to a new host individual. They can also go dormant, right? So they can also hide inside the body of the host, hide from its immune system. So we look across all different viruses, just like when we look across all different life history strategies, there's a wide range of different ones. But same as life history strategies, there's a trade-off. If you're a virus that makes a very severe disease, you've got to have transmission mechanism that's very rapid and effective as well. Otherwise, you're gonna kill the host's gonna die, and all of your virus particles will die, and you won't get to new hosts. All right, so this is for animal cells infected by influenza. When we look at bacteria, there is a range of different things that can happen when they are infected by bacteriophage. Right, so most types of phage can do both of these things. Right, at the top we have what's called the Lytic Cycle. And when a cell gets sort of exploded or disintegrated and the cell membrane goes into little fragments, that's called lysis. Right, so when the cell lyses, all the new phage particles will be released. And so this is often the way that bacteriophage is going to have an infection. It's going to insert the DNA into the host. It's going to replicate like crazy, and it's just going to explode out of the cell, killing it. It can also, however, enter, and depending on circumstances in the host, it may enter this other type of temporary hiding. Right? So it may go sort of dormant, and that's called the Lysogenic Cycle. Right? So the Lysogenic Cycle, it's going to incorporate the DNA into the bacterial genome. It's this little green tidbit in here. Now, every time that cell divides, it is replicating the viral genome, right? So the virus is still getting replicated every time this bacteria divides, right? So it's a strategy that bacteriophage can use to replicate themselves without harming the host cell. At some point, it's going to switch into the Lytic cycle, often when the host is stressed, right? So once the phage feels like its host may die, basically, it wants to get out of the host, right? So it's going to switch into the Lytic cycle. It's going to leave. It's going to get out of Dodge, right? So we can have viral infections that go dormant in all kinds of other eukaryotic organisms as well through different mechanisms. All right, so when we're talking about viral infections, there are some 3,500 or so different types of viruses that are known, and they infect every different kind of living organism. Nobody gets away, just like every kind of living thing has bacteria and fungi trying to infect it, something that's trying to eat it, something that's a parasite on it. There's also viruses. So there are archaeoviruses, there's bacterial viruses, there's eukaryotic viruses. And when we look at all the different types of eukaryotic viruses and we break them down between the metazoa, the plants, the fungi, protists, and then this little group is a weird group because insects and plants share some viruses that the insect is the transmission vector for. So we have all these different types of organisms, and there are some patterns in what kinds of viruses tend to infect them. For example, most bacterial viruses are going to be double-stranded DNA. Most of the viruses that infect eukaryotic organisms are single-stranded RNA plus molecules. So when we look at different kinds of organisms, there's these different patterns in what kinds of viruses they tend to get. All right, so the results of the infection of these different organisms is going to look different in different kinds of hosts, right? So what we look like as animals when we're sick with the virus isn't necessarily going to be exactly the same as what a plant looks like, right? So plants are going to get damage to their leaves, and animals are going to have different symptoms that we'll talk about in a minute. So when we think about this idea of specificity, that each type of virus has a certain species of organism that it can infect, that it has certain proteins, glycoproteins that are going to interact with the cells, where we have among the host organisms closely related species, they may have similar enough receptor proteins that some viruses can infect more than one host. So if we look at influenza, there are a variety of different types. Remember, we call them H1N1, H3N3, because they have different versions of those hemagglutinin and neuraminidase spike proteins sticking up. So where we have closely related species, sometimes there's a mutation that allows that particular flu strain to jump hosts, right? We think of it as being a bird flu, but it has a small mutation that allows it to infect humans, all right? So most of our virus species changes have happened from our domesticated animals. So domestic poultry, domestic pigs, we've gotten a lot of our influenzas from them. All right, so this figure is just showing the variety of different types of influenza, how we think they have gotten from one species to another. The dark arrows are showing where there's been transmissions. So we know that domestic poultry acquired the influenza that infects them from aquatic birds. And so there are these relationships, these evolutionary relationships that we can detect using DNA sequencing or RNA sequencing to find these patterns, or these histories of how the viruses evolved in order to be able to infect different hosts. And the new diseases that humans may get and will continue to get into the future are going to come from other organisms typically. Right, so when we get a viral disease, it's going to have, either we don't care about it because it doesn't have any symptoms, or it's going to cause some kinds of symptoms. And again, we can't tell by looking at the virus necessarily what kinds of symptoms it's going to cause, what types of infection it may cause. So we may have something that we call a common cold. It can actually be caused by a variety of different types of viruses. So what I want you to take away from this slide is that what cells the virus infects in the body, for example, common cold is going to be able to infect epithelial cells of the respiratory system, right? Viruses that cause a pneumonia, infection of the lungs, are going to be able to infect the epithelial cells of the respiratory system. There's other types of viruses that can infect skin cells, right? So we will find them, we will see them, the presence of them, as an infection of the skin, all right? And that we can have particular viruses that cause a variety of different symptoms because they can cause or they can infect a whole bunch of different types of cells. Coronavirus actually turns out to be one of these types of viruses. So it doesn't just infect our respiratory system, it can infect almost any type of cell in the body because it binds to some receptors, MHC1 molecules that are on almost every type of human body cell, and so it can bind to them. So the different symptoms are caused by what happens to the cells that are attacked by the virus. They're going to be damaged and destroyed. And then also the immune system, your own immune system is going to attack these infected cells and kill them. Right? So whatever symptoms we have is going to be caused by either the effects of the virus causing damage to your own body cells, or the effect of your own immune system trying to kill those virus infected cells. Right, the kind of disease is also going to be characterized by how it's transmitted. How does it get from one host to another? We can transmit infections horizontally, that is from one individual to a sort of next to us, another independent individual. When your family all gets a cold, you've caught it from one person, and then they sneeze and cough, and those particles are in the air, and then everybody gets it. There's also vertical transmission, and that's from mother to infant. So a lot of sexually transmitted diseases can be passed from mother to infant during vaginal birth. So transmission is often going to be targeted towards getting the viral particles to the part of the host that they can infect. Sexually transmitted diseases are called that because they infect the reproductive organs. Respiratory diseases are often passed by droplets, by particles that are in the air when you sneeze or cough. Those particles are infecting those tissues, so they're in the mucus, they are airborne, and we get them transmitted to other individuals who are nearby and accidentally inhale them. Blood-borne diseases are transmitted by contact between the blood of an infected individual and an open wound that exposes the blood of a new host. So things like AIDS, it's transmitted by blood-to-blood contact. When we look at the actual viruses that cause these different diseases, there's a whole bunch of different types of structure that they can have. They can, so the actual structure, what these viruses look like, isn't necessarily going to tell us what kind of symptoms they're going to cause. So this is just an example of like, what are the viruses that cause some of these diseases actually look like? What kind of virus are they? Ebola is a filovirus. They're very long and skinny. Rabies are these little bullet-shaped types of viruses. We already talked about influenza and SARS are examples of encapsulated or spherical viruses. And then we have ones, they can be very tiny. Viruses that are similar to one another in structure and genome, dengue virus and Zika virus, for example. Our immune system may be able to, or our vaccines may be able to have some cross-reactivity between those different viruses if they're similar enough. All right. When we look at how many different types of viruses have caused pandemics or become very infected basically populations globally, and how many different, how many people they've killed, there's a long list of different kinds of pandemics. Now, since we invented antibiotic medications, we haven't had any bacterial pandemics. So viruses can't be killed by antibiotic medications because antibiotics kill biota, living things. So bacterial infections like plague and cholera can now be killed by antibiotic medications. However, still, some of the most destructive pandemics in the history of humanity have also been from viruses, such as smallpox and yellow fever, Spanish flu. We've all been talking about that, HIV, AIDS, and COVID here. We don't know exactly how big it's going to be in impact on our human population yet. Right, so this is just kind of interesting and fun. I'm not going to ask you how many people got killed by the Hong Kong flu. All right, so how can we protect ourselves from viral diseases? So viruses, because they are not living things, we cannot use antibiotics in order to kill them. However, they are susceptible, some more than others, to the same kinds of tactics that we use against bacteria. Many of them are killed by boiling or heating, at least, or drying out. They may not be able to live very long outside of a host, several days. They are killed by UV radiation because the capsid particles, UV radiation breaks down molecules rather rapidly. And so the capsids can be destroyed by UV radiation. When we wash our hands, we're actually washing the viral particles off of us so that we don't get them into our internal structures where they can attack living cells. So as we have, as humans, invented better personal protective equipment, PPE, everybody knows what that is, gloves, masks, and other sanitary measures, such as understanding that diseases are caused by microorganisms, bleach, ammonia, just washing with hot water and soap in order to prevent transmission. That's the best way we can protect ourselves from getting the disease in the first place. Virus is the best way we have to protect ourselves, assuming we're eventually going to become infected, because the diseases are out there, is by vaccination. And a vaccination is going to be some way that we can get into the body, usually by an injection. Sometimes they're also inhaled, some component part of the virus particle that your immune system then recognizes, mounts an immune response to, and when you're actually exposed to the real disease, it's able to attack it and kill it before it really makes you sick. So we'll come back to what a vaccine does when we talk about the immune system later on in the semester. But because we don't have very many antiviral drugs, vaccination to prevent a serious infection is the best way that we have to protect our populations from viral diseases. Once somebody does get sick, many viral diseases are not fatal, common cold, it'll still be uncomfortable for a few days. Ones that are more serious, some viruses like rabies, most people don't get exposed to it, so we don't go around vaccinating people from rabies. But if somebody's bitten by an animal that's suspected to be rabid, if we give them the vaccination immediately, they will mount an immune response to it fast enough that even if they've been exposed to it, they will be protected by that kind of last second vaccination that they got. If we just provide supportive care, it turns out that people can survive Ebola if they're given IVs, if they're kept in a hospital situation where the actual symptoms that are made of their body's manifestation of that illness are being treated. So we don't have a way to kill the Ebola virus once a person has it, but if we just keep them alive in a hospital setting, then many more people can survive it than originally were thought. There are some antiviral medications, and the way they work is that they interfere with something the virus needs to do. So there's a handful of different ones, there's not that many, and they don't actually kill the virus, but they do things like interfere with the enzymes that it uses to assemble its capsule, or it interferes with its ability to bind to the host cell. So a lot of these are these sort of protein interactions that it interferes with in order to slow the production of new viruses. Right, so there's still a lot that we don't know about viruses. We don't really know where they came from. There's some hypotheses, either that they started out as cells and then they kind of degraded and they're sort of leftover junk that's self-sustaining, or that they assembled themselves together sort of from scratch, and that we have a bunch of different types of viruses because this happened a bunch of different times. So there's lots more research to be learned about the origins of viruses. And as they continue evolving, our relationship between different viral diseases as humans and wanting to survive them is going to be an ongoing struggle.